Hello. Very good evening to the students who have joined. Can you let me know that if my voice is audible? Also, let me know if you can see the presentation clearly. In the chat box, kindly let me know if the voice is audible to you. Okay, thank you so much for the confirmation. We will wait till it's 6 p.m. and then we will start today's session. Very good evening to the students who have joined. We will wait till it's 6 p.m. and then we will start the session. Meanwhile, please let me know if the voice is audible and you can see the presentation clearly or not. We will start at 6 p.m. So please wait up to then. বাবা দরজাটা লাগায় দাও তো আমার। Okay, so it's 6 p.m. I think we should start with the today's class. Meanwhile, I will just make it open. Okay, so very good evening to the students who have joined. My name is Mousumi Deb and I'm the PMRF Fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm your TA for this NPTEL course of Organic Chemistry in Biology and Drug Development. Today it is our week 12 live session. So today it is our last official live session. We will have one more live session on a day after tomorrow, means on Wednesday from 7 p.m. That will be a summary session. So today it is also more or less a summary session because today we will summarize a few things about the course. But before starting, what I will do is I will give you some questions. I will give you like 12 questions from one from each week of the course content which we have covered till now. I just want to check that if you have remember the topics clearly or if you can answer the questions. Okay, if you have revised the things because last session I have told you already that that was our last session that your examinations are coming over, so you have to start revising the things. So first, what we will do is I will show you like 12 questions, each from one week's course content. So after that, what I will do, I will just repeat few things. Okay, few things which I couldn't cover very well because of the shortage of time. Few things it was not directly involved in your course content, but it's equally important. So I thought it to cover it up for today's session so that you can write your subjective questions in the examinations also. So that we will do, but first, whatever we will do is we will start with the question answer session like always. Okay, I will show you the MCQs and then I will discuss it in detail. I will discuss it in a way so that if you are given some subjective question from that topic, that you can solve easily. So before starting, as always, I will like to point out few things that yeah, there will be a problem solving session every week, and today is the last problem solving session of the Monday session. So the sessions we had from Monday 6 p.m. to 8 p.m. and the sessions are live on Google Meet platform. So if any network disruption happens from my side, please wait for some time. Okay, do not leave the session. I will try to rejoin as soon as possible. There may be some network issues, so don't worry. I will try to rejoin as soon as possible, but do not leave the session. And if you have any problem with your network, you can rejoin at any time. I am recording the session, so that's why I have muted your microphones and camera so that I can record it without any problem. But the chat box is always open. I am monitoring the chat box. Okay, so if you have any doubt, any query, you can let me know through the chat box. Okay, I'm monitoring the chat box every time. So if you have any doubt, any query, you can let me know through the chat box. Otherwise, I will share my email ID at the end of the session. But since it's our official last session, we will have the summary session also. So if you have any problem, any doubt, if you want me to cover up any point, you can let me know by today or by tomorrow morning so that I can include it in the summary class also. Otherwise, I will cover the things in the summary class as my way. But if you have any doubt, specific some doubt or in specific some topic, you can let me know today. I will try to cover it up in our last session, which will be on 16th October from 7 p.m. to 8 p.m. Okay, please try to join that session because that will be our last live interaction session. Okay, so if you have any doubt after the class, please let me know through email, and the presentation and class recording, it will be shared to you. So you don't have to worry if you miss out anything due to the poor network connection. Okay. So as I said, today it is our summary session but before that, I want to see if you have remember the things we have learned so far. So from each week course content, I have given one each problem. Okay. So let's start with the first problem of today's session. So the first problem of today's session is, which statement regarding the drug thalidomide is true? Your options are: number A, R isomer is sedative. Number B, S isomer is sedative. Number C, both R and S isomers can be used as drug. And number D, R isomer is a teratogenic. So you have Identify the statement which is true regarding the drug thalidomide. In the chat box, let me know A, B, C or D, which of the following you think is the correct answer for this problem number 1. Which statement regarding the drug thalidomide is true? Number A, R isomer is a sedative. Number B, S isomer is a sedative. Number C, both R and S isomers can be used as a drug. And number D, R isomer is a teratogenic. I will wait for 2 to 3 minutes, then I will check the chat box. Which statement regarding the drug thalidomide is true? Okay, let me check the chat box now. So I have few answers, number D, number A, number B. So I have three different options as answers. I have number A, B and D, three different options as the answer of first question. So let me check which of the following is the correct answer. So the correct answer for problem number 1 is the statement which is true regarding thalidomide will be it's number A. R isomer is a sedative. Okay, so thalidomide is a very famous drug, or should I say that it was a very famous drug and very commercially very much commercialized drug for regarding during I mean before back in time. So these are the two different structure or two different isomers of thalidomide. Okay, this is the R plus thalidomide and this is the S minus thalidomide. So if you can see the structure of thalidomides, the two structures, I mean, the same. The only difference is the stereochemistry of this position. So only one stereochemistry change in structure can distract, I mean, it can change the complete function of a drug very well. And this is the example thalidomide for this kind of example. Okay, like how stereochemistry can affect the biological effect of a particular drug molecule. Why having a specific stereochemistry is so much necessary. This example is thalidomide is a perfect example for the same. So thalidomide was originally marketed as safe, non-toxic sedative and anti-emetic in the 1950s and rapidly became popular to encounter the effects of morning sickness during pregnancy. Unfortunately, it was instrumental in one of the major medical disasters of modern times when it produced teratogenic effects in developing fetuses and led to babies born with the stunted limbs and other development deformities, the so-called thalidomide babies. As thalidomide was considered such a safe drug at the time, few suspected that it was the cause of the problem, and by the time it was linked to the deformities and withdrawn in 1961, 8,000 to 12,000 babies were affected. Consequently, thalidomide gained a lasting notoriety, which was instrumental in a significant tightening of the regulations surrounding the testing of the drugs. Despite its notorious past, there has been continuing interest in thalidomide and it has become some remarkable properties which indicate a wide variety of clinical uses. Early on, it was recognized that thalidomide has an anti-inflammatory property which was useful in treating leprosy. This activity was eventually linked to thalidomide's ability to inhibit the synthesis of the pro-inflammatory endogenous cytokine TNF-alpha, which is produced by the monocytes. In 1998, the drug was approved for the treatment of leprosy. However, thalidomide has a raft of other properties. For example, it is an immunosuppressant and could possibly be used for the treatment of autoimmune diseases or in countering the immune response to allow organ transplants to be accepted by the host. Interest in thalidomide as an anti-cancer agent started when it was found that it inhibited the angiogenesis by an unknown mechanism. Test shows that thalidomide did indeed have anti-cancer activity, and it entered in phase 3 clinical trials for the treatment of renal cancer and multiple myeloma. On that basis, since then it has been discovered that anti-cancer activities of thalidomide are more complex than its effect on angiogenesis alone. Now, in some patients, thalidomide can boost the immune system by a variety of mechanisms rather than suppress it, and this too may account its anti-cancer activity. As thalidomide can suppress or boost the immune depending on individual circumstances, it is known as an immunomodulator. Thalidomide also appears to arrest the growth of the cells and promote the apoptosis directly. So this is the structure of thalidomide, which I have also mentioned. These molecules are the thalidomide analogs. These are the lenalidomide, and this is the actimide. So analogs of thalidomide have been synthesized with the aim of removing the teratogenic properties. Lenalidomide and actimide, they are two such examples. Both contain an amino substituent on the aromatic ring, which is found to be crucial in producing safer drug. Revlimid, which is this one, entered clinical trials in the year 2000 and was given the orphan drug status in 2001 for the treatment of then incurable disease of multiple myeloma. In 2003, it entered the phase 3 clinical trials and was given the fast track status. It is now approved for the treatment of multiple myeloma. Okay. So this is all about the thalidomide, and it is a very good example to show the importance of stereochemistry in the medicinal chemistry. Okay. So this was our first question. The correct answer was problem number A. Now I will go to the next problem. Problem number 2 says a peptide with 12 amino acids has the following amino acid composition. It has two methionine residues, one tyrosine, one tryptophan, one glutamine, one lysine, one arginine, one threonine, one asparagine, one valine, one cysteine. A specific cleavage of the intact peptide produces the fragment with the following sequence. Okay, these are the sequence which produced by this particular peptide by a specific cleavage and element. Which reagent was used for this specific cleavage? Your options are number A chymotrypsin, number B trypsin, number C V8 protease, and number D pepsin. A peptide having 12 amino acid with the following composition. It has two methionine, one tyrosine, one tryptophan, two glutamine, one lysine, one arginine, one threonine, one asparagine, one valine, and one cysteine. Now, a specific cleavage by a specific reagent produces these fragments. One is the glutamine, cysteine, asparagine, methionine, arginine, then methionine, glutamine, threonine, lysine, tryptophan, and valine and tyrosine. So now you have to identify that which of the following reagent was used for the specific cleavage. Chymotrypsin, trypsin, V8 protease, or pepsin. Identify the correct option. I will give you some time for this question. Think, write on a paper if possible. It's very easy. I know all of you can do it. Which reagent was used for the specific cleavage of the peptide having these 12 amino acids? Which gives us this fragment, these three fragments. Look at the fragments carefully. Okay, let me check the chat box. Okay, I have few answers. Number A. I have only number A as the answer. Okay, let me check if number A is the correct answer for this question or not. So, the correct answer is yes, it's number A. Chymotrypsin is the reagent which is used for this specific cleavage. Okay, so now why climate? So these topics are very important, right? We have learned in I think it was in week first or second week itself that the reagents for the specific cleavage. What is the role of chymotrypsin? What is the role of trypsin, pepsin? All these things you have to remember. Now, because this type of question will be given, I have tried to cover up everything in the particular session which where these topics were. I think it was in week 2. So try to check the previous the uploaded PPT and also practice this kind of problems. So yeah, chymotrypsin breaks the peptide bond involving the aromatic amino acid at the carboxylic acid end. About chymotrypsin, one thing you have to remember that it's adjacent amino acid. There is one restriction. If the amino acid happens to be proline, then hydrolysis cannot take place. Okay, cannot take place. So in the next amino acid, which is connected to aromatic amino acid, happens to be proline, then the hydrolysis won't take place. So if you can look at the fragments carefully, you will see there are true amino acid as we have tryptophan, we have tyrosine at the terminal. This indicates that the reagent which was used, it was specific for the I mean breaking the amino acid which has the aromatic side chain. Okay, and the amino acid which is responsible for the same change is the chymotrypsin. So the correct answer was chymotrypsin. Okay, it was very easy. So this type of question it won't be very difficult. It will be easy, but just you have to remember the particular reagents how it works, and you have to look at the fragments carefully. Then easily you can solve the particular questions. Okay. So now I will go to the next problem. Problem number 3 says, which of the following statements is true regarding SDS polyacrylamide chromatography? Your options are number A, here SDS stands for sodium dodecyl sulfate. Number B, SDS is used to unfold the proteins to make them linear. Number C, SDS binds to proteins non-covalently with a stoichiometry of around 1 SDS molecule per 1 amino acid. Number D, all of the above. Which of the following statement is true regarding SDS polyacrylamide chromatography? Which of the following statements is true regarding SDS polyacrylamide chromatography? Identify the correct statement. Let me check the chat box now. Okay, so I have two answers. Yeah, there will be multiple selective questions also. Okay, there will be MCQs, MSQs, there will be subjective questions of I think 3 to 4 marks, or you have to write the paragraph type of questions. So in the final examination, all type of question you can expect. Okay, so I think I have two answers. Okay, I think multiple answers. I have number D, number B, and number D. So number B and D, two different options for this question I have received as an answer. Let me check which of the following will be the correct answer for this problem number 3. So the correct answer for problem number 3 is, it is number D. All of the above statements are true for the SDS polyacrylamide chromatography. So SDS, yes, it stands for the sodium dodecyl sulfate. So now how it happens, like proteins are folded with positive and negative charge, right? We all know that proteins are folded in positive and negative charge, and we also have the disulfide bonds in the protein. So when the protein is by particular disulfide bonds are reduced by two ethanol markaptothiol. So these disulfide bonds are reduced, and we have this two thiol new moieties. Okay, and SDS with the negative charge, what it does do, it binds with this particular protein and unfolded linear structure with a negative charge potential to the polypeptide chain length. Okay, so based on the chain length, it binds with the particular SDS which has a negative charge, and then if we have this particular molecular weight markers, so the based on their molecular weight, the particular chains they become separated. So purification of proteins by SDS page, it is also known as SDS page. Polyacrylamide gel chromatography, also it's known as. So the technique involves loading the sample of your mixture onto a polyacrylamide gel, which is the short form is page. Polyacrylamide works like agarose, except the matrix is a smaller pores, and so the polyacrylamide gel separate the smaller molecules like proteins. Agarose is used for the much larger molecules like DNA and RNA. I have mentioned it the use of the agarose gel already when we were discussing the solid state DNA synthesis. Unlike DNA and RNA proteins, the proteins do not have any nice constant charge to mass ratio and can have any charge at a given pH depending on the sequence, hence PI. To overcome its problem, proteins are coated with the detergents SDS, which makes them negatively charged. They then separated by the molecular weight. The SDS will disrupt the secondary, tertiary, and quaternary structure, so the subunits will separate. For this reason, the SDS page separates by subunit molecular weight. So there we have learned many separation techniques, like we have learned that SDS page chromatography. Then we have discussed many chromatography like column. We have discussed the charge exclusion chromatography and so on. So these purification techniques can be given like in the question in the examination can be given like write a short note on SDS page or any kind of technique. Okay, purification technique. So this type of question you can expect in the final examination. So yeah, problem answer number D was the correct one. Now I will quickly move to the next problem. Problem number 4 says, which one of the following statement is true regarding the thiol proteases? Your options are: number A, it can react only via catalytic triad of aspartate, histidine and cysteine. Number B, histidine is not necessary in this protease. Number C, aspartate is the most important in this catalytic triad. Number D, histidine is significant to activate these proteases because of the more nucleophilicity of sulfur. Identify the statement which is true regarding the thiol proteases. Again, I will repeat the options. The first option is it can react only via catalytic triad of aspartate, histidine and cysteine. Number B, histidine is not necessary in this protease. Number C, aspartate is the most important in this catalytic triad. And number D, histidine is sufficient to activate these proteases because of the more nucleophilicity of sulfur. Identify the true statement regarding the thiol protease. Which one of the following statement is true regarding thiol protease? Let me check the chat box. The final examination date is given as 26 October, as per I know, in the portal, right? In the SWAM portal. If there is any change in the date, anyways, you will be notified regarding the examination by, I think, 17th of October, you will be notified the exact date. Okay, I think it is on 26th October. Okay, so for the previous question, I think I have few answers. I have number D as the answer. Okay, let me check if number D is the correct answer or it's not. So the correct statement regarding thiol proteases is, yes, it is number D. Histidine is sufficient to activate these proteases because of the more nucleophilicity of sulfur. So this is basically an example how the thiol proteases work. So basically in the thiol proteases, it uses the thiol as the nucleophile and we have a histidine. So in the histidine it has a particular heterocyclic ring which is known as the imidazole ring, right? It is a 5-member ring and it has 2 nitrogen in 1 and 3 position. So this nitrogen will use its lone pair to take up this proton from the thiol and it makes the sulfur more strong nucleophile, and this nucleophile nucleophile increase increasing this more nucleophilicity of sulfur, it will attack to the carbonyl carbon of the peptide linkage, and it will make a tetrahedral intermediate like this. Now, when this tetrahedral intermediate, this oxygen reverses back to make this carbonyl carbon, and this bond breaks, and it takes up the proton from the earlier protonated imidazole ring of the histidine. Okay, and then we have this two different fragments. So this thiolated fragment, it is then there is a water which will hydrolyze this thioester of this peptide bond, and this thioester will be hydrolyzed by this water, and it will give us the carboxylic acid of this particular portion, and the thiol and this imidazole ring, which we will be same as before of the catalytic cycle. So this is the complete catalytic cycle. So histidine is sufficient to activate this proteases because the sulfur has a higher nucleophilicity than that of the alcohols. Okay. So option number D was the correct one. Now I will move to the next problem. Problem number 5 says the restriction enzyme ALUI cleaves between the base pairs of, your options are number A and T, number B G and C, number C A and C, and number D G and T. Which of the following base pairs are cleaved by this restriction enzyme ALUI? The restriction enzyme ALUI cleaves between the base pair of number A and T, number B G and C, number C A and C, and number D G and T. The restriction enzyme ALUI cleaves between the base pair of which two base pairs? Okay. Number A, adenine and thymine. Number B, guanine and cytosine. Number C, adenine and cytosine. Number D, guanine and thymine. We have discussed about the different restriction enzymes already. So these topics are also very important. The restriction enzymes, which enzyme cleaves where, these things you have to remember. Okay. Which enzymes produce the sticky ends, which enzyme produce the blunt ends, all I have discussed in earlier in the week 5 course content, I think it has. So you can check the respective presentation which I have supplied. So the restriction enzyme ALUI cleaves between which base pairs? Let me check the chat box. Okay, I have answers like number B, number C. So I have two different options. One is number B and another is number C as the answers. Let me check which of the is the correct answer. So the restriction enzyme ALUI cleaves between the base pair number B, G and C. Okay, so it cleaves between the guanine and cytosine. So restriction enzyme that cuts DNA at specific nucleotide sequences known as the restriction sites. It is found in the bacteria and have evolved to provide the defense mechanism against the invading viruses. In bacteria, they selectively cut up the foreign DNA in the process called the restriction. To cut the DNA, restriction enzyme makes two incisions each strand of a DNA double helix. So ALUI, it cuts between the G and C. Okay, and HA it cuts between the G and C again. Then both of these produce the blunt ends. So why these are called the blunt ends? Because they are cutting at the same position of the 5 prime to 3 prime and 3 prime to 5 prime, two strands. Okay, but BMHI, this cuts between the G and G, but this produce the sticky ends. Why sticky ends? Because as you can see here, it cuts in this position, but here it cuts this position. Both the positions are not same in the case of the two strands. HIND triple I, it's also produce the sticky ends, and it cuts between the A and A, means adenine, and Ecori, it cuts between the G and A, and it's also produce the sticky ends. Okay, so which restriction enzyme cuts where and which produce the blunt end, which produce the sticky ends, these things are important. Okay, so you have to remember this point. So yeah, the correct answer was number B. Now moving toward the next problem. Problem number 6 says, in the chemical synthesis of DNA, 5' OH is protected and deprotected by, your options are number A, dimethoxytetracyl group and ammonia, number B, benzoyl group and ammonia, number C, benzoyl group and tetrazole, number D, dimethoxytetracyl group and dichloroacetic acid. In the chemical synthesis of DNA, 5' end is protected by which reagent and deprotected by which reagent? You have to identify the reagents respectively. Means 5' end is protected by either number A, dimethoxytetracyl group and deprotected by ammonia, or number B, it is protected by benzoyl group, deprotected by ammonia. Number C, it is protected by benzoyl group and deprotected by tetrazole. And number D, it is protected by dimethoxytetracyl group and deprotected by the dichloroacetic acid. Identify the correct answer. In the chemical synthesis of DNA, 5' end is protected, deprotected by which chemical groups? Identify the correct option. I will give you 1 more minute and then I will check the chat box. A, B, C or D, which of the following is the correct answer? All these things we have already discussed. I just want to revise the things so that we can, this will be the last problem solving sessions like this. Then I will just summarize the few things from the course. So try to answer. Okay, let me check the chat box. Okay, I have few answers. Number D, number D. Okay, so it seems like number D is the only given answer for this question. Okay, let me check if number D is the correct answer or not. So the correct answer is yes, it's number D, dimethoxy, trityl group, and dichloroacetic acid. In the chemical synthesis of DNA, 5' OH is protected by DMT group and it is deprotected by dichloroacetic acid. So already I think I have discussed this particular chemical synthesis of DNA, that it is the your deoxyribose sugar. This is the base in the 1' position, and in the 5' position we have this alcohol which is protected or blocked by the DMT group. Okay, and this in the 3' position it is there is a phosphoramidate linkage, and there we have this diisopropyl amino group. So nucleoside phosphoramides are used to chemical synthesis of DNA. Nucleotides are modified to ensure that the correct group reacts with the next reagent. Each nucleotide has a DMT group blocking its 5' OH, and 3' OH is activated by a phosphoramidate group, which is originally also protected by the diisopropylamine. Okay, so this particular 3' group is activated by this phosphoramidate group, and it is also protected, which is in general protected by this diisopropylamino group, and the next nucleotide will join here. The next nucleotide it should not join at the 5' position. That is why the 5' position is blocked by the DMT group, and this DMT group means this one. Okay, as you can see, it is the dimethoxytrityl group. This is the DMT group structure, and it is deprotected by the dichloroacetic acid. Okay, so this is the particular, or it can be also deprotected by the trichloroacetic acid. Both the dichloro and trichloroacetic acid can be used to deprotect this DMT group. Okay, so option number D was the correct one. Now I will go to the next problem. Problem number 7 says the hydrogen that are most likely to get exchanged with the deuterium when the following molecule is dissolved in D2O. Your options are number A, HA only, number B, HB and HD, number C, HB and HC, and number D, HD only. So which of the following hydrogen in this molecule? There are 4 hydrogen. This is HA, this is HB, this is HC, and this is HD. So you have to identify that which hydrogens are most likely to get exchanged when with deuterium when the following molecule is dissolved in D2O. So if the following molecule is dissolved in D2O, which of the following hydrogen is most likely to get exchanged? Identify the hydrogens which are most likely to get exchanged when deuterium when the following molecule is dissolved in D2O. This is HA, this is HB, this is HC, and this is HD. Okay, let me check the chat box. Okay, I have few answers. Number C, number A, number B, number A. So I have 2 number A and 1 number B, 1 number C. So I have A, B, and C. So different answer for this question. Let me check which of the following will be the correct answer for problem number 7. So the correct answer means the hydrogens which are most likely to get exchanged when deuterium, when the molecule is dissolved in D2O is or are number B and number C. Okay, means it is the HB and HC, means option C is the correct answer. This proton and this proton, these two protons will be most likely to dissolve in D2O. So actually, if you look at carefully the structure, okay, this proton HC will be the most likely to exchange because this is the alcohol proton. Okay, so this alcoholic proton has the most tendency to get detached, okay, and to get exchanged. Now this, because this is the most acidic. Now if you look at the HB, this exists between two heteroatoms, means one nitrogen and sulfur, which makes this proton more acidic, and that's why the rate of exchange of this proton will be higher. Then HA and then HD will be the last one because it is the aromatic proton, which is not so easily, the aromatic protons are not very easily get exchanged with the deuterium. Okay, so the rate of exchange will be higher for HC, then HB, then HA and HD. So HC and HB are the most readily get exchanged with the deuterium. Okay. The deuterium exchange peaks that are due to the proton OH and NH protons can be identified by the exchange of deuteriums. Okay, so this deuterium exchange is a very good technique to identify the OH or NH protons. Deuterium is not observed in the proton 1 NMR spectra. Shaking a sample containing exchangeable proton with the D2O causes the exchangeable proton to be replaced by deuterium, and as a result, the signal for the exchangeable proton disappears and from the spectrums. Okay, so this is the alcoholic OH with the D2O. It is converted to ROD, and with the HOD we get. Okay, so easily this alcoholic and NH protons get exchanged with the deuterium, and whenever in the proton spectrum the peak for the alcoholic OH or NH, it will be disappeared if it is getting exchanged with the deuterium, because deuterium do not show any peak in the proton NMR. So hydrogen deuterium exchange in NMR spectroscopy, one way to testing if the peak is from OH and adding a drop of deuterated solvent D2O and MEOD. Because the exchange of deuterium, the peak disappears since the deuterium is NMR inactive at the proton's frequency. Okay, the deuterium, it is very important to remember that the peak disappears because deuterium is NMR inactive at proton frequency. Okay, deuterium is not. It is itself is not. It is inactive in proton's frequency. So as you can see here, we have the OH peak in this position, but when it is replaced by this deuterium or exchanged with the deuterium, there will be no OH peak. Okay, so this is the example. Now this is the technique to identify if there is this kind of acidic OH or NH protons in a sample. So now I will move to the next problem. Problem number 8 says, which of the following is the correct order of the IC50 values of the following anti-cancer agents, anti-agents against the human cancer cell lines? Your options are number A is the pyrimethamine, number B is the trimethoprim, number C is the methotrexate. So you have to identify the correct order of IC50 values of the following agents. Your options is A greater than B greater than C, number B is B greater than C greater than A, number C is B greater than A greater than C, and number D is C greater than A greater than B. A is the pyrimethamine, number B is the trimethoprim, C the methotrexate. So you have to identify the correct order of the IC50 values of the following anti-cancer agents. Which of the following is the correct order of IC50 values of the following anti-cancer agents? Let me check the chat box. Okay. So don't worry, you will not be given any answer like where you have to draw anything on computer. Okay, so you don't have to worry about it. Okay, I think you will be given such as you have to write on a paper and then upload. If in case of any structural questions are asked, there will be option to upload an answer, the written form. Okay, so be ready with the paper or pencil or the pen so that you can write, you can click a picture and upload for the same. Okay. So I think I have few answers. I have number A, number C as the answer for the problem. Okay, let me check if the which of the following is the correct answer. So the correct answer is it is number C. The IC50 value of trimethoprim is greater than pyrimethamine and the lowest IC50 value is of the methotrexate. So IC50 is the half maximal inhibitory concentration, is a measure of the potency of a substance in inhibiting a specific biological or biochemical function. IC50 is a quantitative measure that indicates how much of a particular inhibitory substance or a drug is needed to inhibit in vitro a given biological process or biological component by 50%. The biological component could be enzyme, cell receptor, microorganism. IC50 values are typically expressed in the molar concentration. So all these molecules are acting as the drugs. For example, pyrimethamine acts on the plasmodium and is reflected in the acidity value. Acidity value is the inhibitory concentration. As I have already mentioned, it is required to inhibit the 50% of the activity. So acidity value of the plasmodium is in the bacteria in 0.05 microgram per milliliter. In the bacteria is 250 and the minimal mammal is the 70. So if it is extremely specific for plasmodium, okay, so this pyrimethamine I am talking about. So pyrimethamine is used as an anti-malarial drug because it is organic from malaria. So it is the anti-malarial agent. Trimethoprim is a bacterial dihydrofolate reductase inhibitor. The IC50 value of the bacteria is 0.5 and plasmodium is 7. So it is 10 to 14 times higher. Remember, the lower the IC50 value, stronger is the potency for the mammal and totally ineffective because IC50 value is to 26,000. So one of the use of the one can use this trimethoprim as an antibacterial agent. So methotrexate IC50 value is 0.1. Okay, now for the bacteria and plasmodium in mammal it is 0.02, so it can act as an antibacterial agent, antiplasmodium agent, and also it can act as a dihydrofolate reductase in mammal. It kills the cells in human body, specifically the cells which grows very fast, that is the cancer cells. So methotrexate is used as an anti-cancer agent. Okay, this is the this is the antimalarial agent. So this particular methotrexate mainly used as an anti-cancer agent because it has the lowest IC50 value in the human cancer cells. But the IC50 values for this pyrimethamine and trimethoprim, these are much greater in the cancer cells. So these cannot used as a particular anti-cancer agents. Okay, pyrimethamine is mainly used as a anti-malarial agents, whereas this trimethoprim is mainly used as the antibacterial agent. Okay. So now problem number 9, it says when a drug is approved for the human testing during the clinical trials. Your options are number A, between phase 3 and phase 4, number B, between phase 1 and phase 2, number C, at the end of phase 4, and number D, at the end of the preclinical trials. When a drug is approved for the human testing during the clinical trials. Your options are number A, phase 3, Number A is between the phase 3 and phase 4. Number B between the phase 1 and phase 2. Number C is at the end of the phase 4, and number D at the end of the preclinical trials. When a drug is approved for the human testing during the clinical trials. Number A between phase 3 and phase 4. Number B between the phase 1 and phase 2. Number C at the end of the phase 4, and number D at the end of the preclinical trials. So when a drug is approved for the human testing in during clinical trials, let me check the chat box now. Okay, I have several answers. Number C, then I have number D. So I have, yeah, the date of the examination may be on 26th or 31st. It will be notified you. I have already mentioned it will notify you. And yeah, there will be subjective question given in the examination. Okay, not only objective, subjective, it will be a mixed type of questions. So I have two options, number D and number C as the answer for this problem. Let me check which of the following is the correct one. So, a drug is approved for the human testing during the clinical trials at the end of the preclinical trials, which is the correct answer is number D. Okay. So this is the clinical trial process. I have already mentioned this is a very important chart which you have to remember. The total duration of a complete clinical trials is 5 to 15 years. Specific designation as speed up to process can speed up the process like some emergency like COVID or pandemic. It can speed up this process, but in general, clinical trial process is up to 5 to 15 years. So first is the preclinical, exploring if and how the new drug may work. Phase 1 is the exploring safety and dosing of the drug. The drug is approved from human testing in between the preclinical trial and phase 1. Okay, so at the end of the preclinical trial, the drug is approved for the human testing. Okay, then we have the phase 1 when the exploring the safety and dosing of the drug. In the phase 2, exploring the effectiveness of the drug. Phase 3, exploring the safety and effectiveness of compared to the currently available treatments. Then we have the FDA review to confirm the safety and effectiveness of the drug. Okay, in between the phase 3 and FDA review, the drug is submitted to the FDA approval at the end of the phase 3. Then we have a phase 4, which is also known as the post marketing surveillance, which is the evaluating the drug over a time of the large number of patients. Okay. So after the FDA review, the drug is approved. When then it is checked after a few years to see if in the larger number of patients it has any particular kind of side effect or not. So this is the total duration and total specifications of the clinical trial process. Okay. Now moving to the next problem. Problem number 10 says sulfonamides are dash inhibitor of the dihydrofolate synthetase. Your options are number A reversible, number B irreversible, number C suicide, and number D ASMII. Okay. So sulfonamides are dash inhibitor of dihydrofolate synthesis. Sulfonamides are dash inhibitor of dihydrotoroid synthetase. Number A is reversible, number B irreversible, number C suicide, and number D ASMII. Sulfonamides are dash inhibitor of dihydrotoroid synthetase. Is it reversible? Is it irreversible? Suicide or ASMII? Which type of inhibitors are sulfonamides? Let me check the chat box now. Okay, so I have few answers. Number C and number B are the given answer. Let me check which of the following is the correct one. So the correct answer for problem number 10 is number A, reversible. Sulfonamides are the reversible inhibitor of dihydrotoroid synthetase. Okay, so this is the sulfonamides. They are the reversible inhibitor of dihydrotoroid synthesis. Okay, so this is the dihydrotoroid. This is the structure, and we have this para-aminobenzoic acid as the substrate. Okay, now then we have this from dihydrotoroid. We have the dihydrofolate, and then we have the tetrahydrofolate. So this is the conversion already which I have mentioned. This is the mechanism of action of the sulfonamides, how sulfonamides work, and this is the synthesis structure of the tetrahydrofolate or coenzyme F. Okay, so particular the sulfonamide, it inhibits reversibly this dihydrotoroid synthesis. Okay, which is the which actually converts this phosphate, particular phosphate, to this dihydrotoroid, and then we have this from dihydrotoroid we have dihydrofolate, and from dihydrofolate we have the dihydrofolate reductase enzyme which converts the dihydrofolate to tetrahydrofolate, and the trimethoprim, it inhibits this particular dihydrofolate reductase, and sulfonamides, they inhibit the dihydrofolate synthetase. Okay, the reversible inhibition was the correct answer. Now problem number 11, it says that acyclovir is an inhibitor of DNA polymerase selectively in the virus infected cells. This is because number A, acyclovir is first converted to monophosphate only by the host thymidine kinase. Number B, acyclovir does not enter the infected cells. Number C, acyclovir is first converted to monophosphate only by the viral thymidine kinase. Number D, acyclovir triphosphate does not accepted by the host DNA polymerase. Which of the following statement is correct regarding the acyclovir, which is an inhibitor of DNA polymerase selectively in the virus infected cells? It is because number A, acyclovir is first converted to monophosphate only by the host thymidine kinase. Number B, acyclovir does not enter the infected cells. Number C, acyclovir is first converted to monophosphate only by the viral thymidine kinase. Number D, acyclovir triphosphate is not accepted by the host DNA polymerase. Acyclovir is an inhibitor of DNA polymerase selectively in virus-infected cell. What is the reason? Let me check the chat box now. Okay, I have options like number C. Okay, let me check if number C is the correct answer or not. The correct answer is yes, it's number C, acyclovir first converted to the monophosphate only by the viral thymidine kinase. So this is a very important example. So how the selectivity of the drug targets can, I mean, contribute to the particular selectivity of a drug, because the DNA polymerase, the structure is different than the viral DNA polymerase, the human and viral. So this difference of the structure can be exploited, like in the example of this. So this is the acyclovir. This is a prodrug. Okay, acyclovir is a prodrug. So there are two different kinases. One is the cellular thymidine kinase of the host, and another is viral thymidine kinase. And so this viral thymidine kinase, it phosphorylates this particular acyclovir, and then cellular thymidine kinase can make this triphosphate, which is the active drug. Okay. Acyclovir is first converted to monophosphate by viral thymidine kinase. This step makes this give the selectivity of the acyclovir towards the virus infected cell. Okay, the first phosphorylation does not happen with the cellular thymidine kinase. It only can be done by viral thymidine kinase, and then followed by the di and triphosphorylations can be done by the cellular thymidylate kinase. Okay, and this P examples are the phosphate. This is the acyclovir. This is the prodrug. This is the structure of the acyclovir, and this is the acyclovir triphosphate, which is the active drug. And the activation is initiated by the viral thymidine kinase. That is why only virus-infected cells this acyclovir works. Okay, not the normal human cells. So now last question. Problem number 12, it says that cyclophosphoramide is commonly used in anti-cancer therapy. Which of the following statements is false regarding its action? Your options are: number A, it is itself a bisalkylating agent. Number B, it acts as a prodrug. Number C, it needs oxidative activation to show bisalkylation activity. And number D, the structure is metabolized to release the acrolein. Cyclophosphoramide is commonly used in anti-cancer therapy. Which of the following statement is false regarding its action? Number A, it is by itself a bisalkylating agent. Number B, it acts as a prodrug. Number C, it needs oxidative activation to show bisalkylation activity. Number D, the structure is metabolized to release acrolein. Okay, let me check the chat box. Okay, I have two answers, number C and number A. Let me check which of the following is the correct answer. So the correct answer is number A. It itself, it is by itself a bisalkylating agent. Okay. This statement is false regarding its action. So cyclophosphoramide is an alkylating agent. It is widely used as a DNA cross-linking and cytotoxic chemotherapeutic agent. It is given orally as well as intravenously with efficacy. It is inactive in parent form but must be activated to cytotoxic form by the liver CYT450 liver microsomal system to 4-hydroxycyclophosphamide and allophosphoramide. The 4-hydroxycyclophosphamide and allophosphoramide are delivered to the delivering normal and tumor cell. Okay. So cyclophosphamide, the mechanism of action, the main effect of cyclophosphamide is due to its metabolite phosphoramide mustard. The phosphoramide mustard forms DNA cross-link between the intrastrand linkage and within the intrastrand link cross-linkings. DNA strands at 1 in 7 position is eventually leads to the cell death. So this is cyclophosphamide. It can lead to the off-target oxidation also. Then we have CYP450 enzymes which converts this to 4-OH cyclophosphamide. Okay. Which has a hydroxyl in the 4 position. Then we have the allophosphamide, which linkage is broken, and we have a aldehyde and amine. These are responsible for the detoxification, and the allophosphamide is related to the acrolein, which is for tissue toxicity, and this also gave rise to the phosphoramide mustard, which leads to the DNA and protein alkylation and cell death. Okay. So these were the 12 questions for the 12 weeks which we have already covered. Okay, so this now what I will do is I will cover few topics which are not which will not be only the question answer. Now what I will show you, it will be a little discussion of the topics which I think are important. I want you to revise these things, and also there were few topics which I got questions like for example, somebody has told me to cover up on little bit about the cholinergic receptors. So that also I will do in today's class. Okay, and also I will discuss about some inhibition of the drugs and so on. So now let us start with the revision portion. Okay. So when we started our course, the first thing which we discussed were the amino acids. Okay, so we have 9 essential amino acids. These things are very basic, but I want you to revise these things. Okay, so there are 9 essential amino acids among all the 20 amino acids. This 9 essential amino acids, this we have to take up from the food because these are not synthesized in our body. That is why they are known as the essential amino acids. Many commercial you will feel of the food products which have the 9 essential amino acids. So what are the 9 essential amino acids? We have the phenylalanine, we have valine, threonine, tryptophan, methionine, leucine, isoleucine, lysine, histidine, and histidine. So phenylalanine is crucial for the neurotransmitter production, impacting the brain function and mood regulation. Valine, important, crucial for the muscle repair, growth, energy, blood sugar, and immune function. Threonine, which is important for the bodily function, nervous system, health, collagen, elastin, and tissue maintenance. Tryptophan is crucial for the serotonin production, mood regulation, and niacin synthesis for the energy and cellular health. We have methionine, which is crucial for the methylation and as a precursor for the antioxidant and detoxification. We have leucine, which is crucial for the muscle growth, repair, and regulating blood sugar, providing the exercise energy. Isoleucine, crucial for muscle repair, endurance, and the regulating blood sugar and during exercise. Lysine is crucial for the growth, tissue repair, supporting healthy immune system, and histidine. This is crucial for histamine production and precursor, a key component in physiology. So these are the 9 essential amino acids. All the structures are given here. So I will suggest to remember some structure of the amino acids also, because the questions can be given from amino acid, which can all easily can be done if you remember the structures of the amino acids, especially the amino acids which contain acidic side chain, basic side chain, aromatic heterocyclic side chains, and so on. Okay, so these remembering the amino acids are very important. Then we have the classification of amino acid regarding the polarity of the side chain. Okay, so polar amino acids where the R contains the polar hydrophilic group, so it can form the hydrogen bond with water. In those amino acids, R may contain a OH group as in serine, threonine, tyrosine. It can contain SH groups such as cysteine. It can contain amide group as in glutamine and asparagine. It can contain NH2 group or nitrogen act as a base in basic amino acids such as lysine, arginine, and histidine. And carboxylic acid group in acidic amino acid, the aspartate and glutamic acid. So acidic, basic amino acid, these are also polar amino acids. The neutral amino acids like serine, threonine, all these are the polar amino acids because they have a polar side chain which are capable of forming the hydrogen bond with water. We have non-polar amino acids also, like R is the alkyl group or hydrophobic group, which cannot enter in the hydrogen bond formation. 9 amino acids are non-polar, such as glycine, alanine, valine, leucine, isoleucine, phenylalanine, tryptophan, proline, and methionine. These amino acids are non-polar because these have the alkyl hydrophobic group, which cannot enter the hydrogen bond formation. 9 amino acids, the examples are all these. So this remembering which amino acids are polar, which are non-polar is also very important. Then we have the classification of proteins as the fibrous and globular protein. So fibrous proteins, as I already mentioned, it is a class of insoluble protein which constitute the main structural elements of our body. Okay, the fibrous protein, they constitute the main structural elements of our body, such as the skin, hair, all these are the structural proteins. Okay, and the globular protein, a protein soluble in water and tend to involved in the metabolic function. These are the mainly the enzymes which are involved in the metabolic functions. These proteins are the globular protein. Okay, and these are the water soluble, but the structural proteins or the fibrous proteins are not water soluble, such as hair, skin. As you can see, our hair or skin, they are not soluble in water, right? Because these are the made of the fibrous protein which are insoluble in water. Fibrous protein has a long and narrow shape. Globular protein, as the name suggests, it has a round and spherical shape. Fibrous protein has the helical or sheet structure. These are the secondary structure which we have already discussed. And globular protein has a folded like a ball-like structure. Now, the fibrous protein are mainly the secondary structure of protein, mean the alpha helix and beta pleated sheets. And that globular proteins are mainly the tertiary structure. Now, the tertiary structure, it forms when the beta pleated sheet or the alpha helix, they interact with each other via various interactions like disulfide bond formation. We have the pi-pi stacking, which has salt bridge kind of interaction, and so on. So whenever the sheets and the helix, they interact with each other, they forms this particular tertiary structure which has a spherical round like shape. And the fibrous protein has a repetitive amino acid sequence. Globular protein consists of an irregular amino acid sequence. Fibrous proteins are insoluble in water, acids, and bases, and globular protein are soluble in water, acid, and bases. Fibrous proteins have strong intermolecular interaction. Globular proteins have weaker intermolecular interaction. Fibrous proteins are less sensitive to the charges in the changes of the temperature and pH, and globular proteins are sensitive to the changes in temperature and pH. The fibrous proteins are involved in forming the structure. Globular proteins are involved in the metabolic reaction. Example of fibrous proteins are collagen, elastin, actin, myosin, fibrin, keratin. All these are present in our skin and hair. You have heard of the collagen and elastin on our skin, keratin for the hair also. So these are all example of the fibrous proteins, enzyme, hormones, hemoglobin, immunoglobin, which are involved in the various metabolic functions in our body. These are the globular protein. Okay. Then we have the very important thing is the Zwitterion and isoelectric point. The calculation of isoelectric points are very important. I have already mentioned it clearly in the first week slides. Okay, when you are revising, go to the first week's PPT which is uploaded. I have shown you how to calculate the isoelectric point for a neutral amino acid, acidic amino acid, and basic amino acid. All these things are discussed in detail. Mainly, you have to remember the particular amino acid in the physiological condition. This exists in the zwitterionic state, such as amino group present as NH3, carboxylic acid present as the COO minus. Okay, so zwitterion means plus and minus, both charge are present in the one single molecule. Each amino acid has an isoelectric point or pi, which there is a no net charge. Okay, for most of the amino acid, isoelectric point is zwitterionic state, but in another amino acids such as aspartic acid, arginine has an isoelectric point which favors more acidic or basic form. Okay. So pH dependent structure of the amino acids with the acidic side chain. So when the particular amino acid is neutral, okay, like for example glycine, it will have only two pKa values, one from the amino group, one from the carboxylic acid group. And the calculation of the isoelectric point means finding out the pH when it will be exactly neutral. It is very easy. You have to just take up two pKa values. You have to average it out. Okay, that is about how you will identify what is the PI or isoelectric point of a normal neutral amino acid. But when the amino acid becomes acidic and when the amino acid became basic, the calculation it changes. Okay, so for an acidic amino acid, it will have two different carboxylic acids. So it will have three pKa values. The first pKa value is for the alpha COOH, okay, alpha carboxylic acid group. So when the initially the net charge is +1, then when the first amino acid is first carboxylic acid group, it is get decarboxylated. We have this new net charge 0. That the pK2 is for the side chain carboxylic acid group and the pK3 is for the amine group. So the change of the net charge is goes like this: +1, 0, -1, and -2. It is for the acidic. So for an amino acid having an acidic side chain, the positively charged form +1 dominates at a very low pH. The zwitterionic neutral form dominates at a low pH, and the negatively charged form or -1 form dominates the intermediate pH. And at strongly basic pH, doubly negatively charged form -2 may form, which is this one. So isoelectric point or PI should average the pK values of the two most acidic groups, or that is pK1 and pK2, because in between this pK1 and pK2, as you can see, these two steps, it is converting from +1 to -1. So when you are averaging these two pK values, you will have the isoelectric point of the acidic amino acid. So in short, you have to average the two lowest pK values for in the case of acidic amino acid. Okay. But what is for the basic amino acid? So in the basic amino acid, we have a carboxylic group, we have acid group, we have a NH3 group, which are the alpha groups. But we have in the side chain, we have a extra amine group. So in the strong acidic pH, it has a particular net charge of +2 when all the groups are the protonated. Then after at the first pK is for the alpha carboxylic acid. What it will will happen? So when the first pK is there, so at this pH, this group will be deprotonated, and we have the net charge of +1. In the second pK2 value, this group pK2, this amino group, alpha amino group will be deprotonated. We will have the net charge 0, which is at the basic pH, and the very strong basic pH, we will have a net charge of -1. So here the PI value will average out the two most basic group, the pK2 and pK1, pK3, because in between the pK2 and pK3, this has a net charge of 0. So when there is a basic amino acid, the doubly positive charge +2 dominates as a very low pH. The positively charged form +1 dominates at the intermediate pH. The zwitterionic form or neutral forms at the basic pH. So at the strongly basic pH, the negatively charged form -1 dominates. Okay. So you have to remember that whenever there is a basic amino acid, the isoelectric point or PI calculation will be like you have to average the two higher pK values, begins pK2 and pK3. Okay. So for neutral, only two are there, so you have to average the two. In acidic, you have to average the lowest one, or pK1 or pK2, and in case of basic, you have to average the more basic pKs, means the pK2 and pK3. Okay. Now, enzyme inhibition, we have discussed a lot, I think, when we are having our normal class, how the reversible enzyme inhibitions happen. So, all the graphs, okay, so the normal Michaelis-Menten kinetics, you have to read. You have to remember the derivation of the normal Michaelis-Menten kinetics. I have not mentioned it here because I already have mentioned it in our regular classes. Okay, you can check it from there. The normal enzyme inhibition for using the Michaelis-Menten kinetics, how it happens. Okay, so there are different kind of enzyme kinetics, right? So whenever before starting this inhibition, just let me tell you the things that whenever you are reading the enzyme kinetics, the most important things which you have to remember is the graphs. Okay, the graphs, the Michaelis-Menten graph, the Lineweaver-Bark plot, the Hansful plot, all the graphs you have to remember. What what is in the x-axis, what is in the y-axis, how the graphs are coming from the Michaelis-Menten equation itself. I have already discussed it in the in the presentations. Okay, it was in the, I think, week 2 or week 3. I don't remember exactly, but I have discussed it. And from the Michaelis-Menten, these things you can expect few mathematical questions also. So practice these things. The questions will be easy, but you have to practice. Otherwise, you won't get the answers correctly. Okay. Also, when we were discussing the enzyme kinetics, one thing we have discussed was the enzyme inhibition. So I didn't discuss the enzyme inhibition kinetics that time because it was not very deeply included in your course. But I think it is important for you to understand how the different Lineweaver-Burke plot of the inhibition comes from, because I have only shown you how the graphs look like in case of competitive, uncompetitive, and reversible, irreversible, all these things. But how this graph comes from and what is the kinetics, let us discuss a little bit. Okay. So the first type of inhibition which we are going to read is the reversible inhibition. So many inhibitors reversibly binds with the enzyme molecule, affecting the equilibrium constant of the reaction. Several types of such inhibitions are known and can be distinguished with the help of the Lineweaver-Burke plots of the enzyme kinetics of the three subtypes. So first reversible inhibition is the competitive inhibition. So all these are the reversible inhibition which I am discussing. Okay. So this reversible inhibitors bind with the enzyme reversibly, and okay, so it does not permanently change the particular enzyme's action. Okay, it reversibly binds and reversibly it can affect the enzymes, but it do not permanently damage the enzyme. So first type of reversible inhibitor is the competitive inhibitor, and the competitive inhibition is inhibitor is a structurally related substrate. It competes with the substrate for the active site. So we all know that enzyme has an active site, okay, where the structure of the active site is such that a particular only the substrate molecule can bind to the active site. But competitive inhibitors are the molecule which are structurally very similar to the particular our substrate molecule, and it can perfectly bind to the active site of the enzyme, and it binds reversibly at the active site of the enzyme to form the enzyme inhibitor complex, EI. Okay, enzyme inhibitor complex means EI complex. So as you can see the equation, this is the enzyme plus substrate, it forms the enzyme substrate complex. The initial forward rate constant is K1, backward is K minus 1, and we have this enzyme substrate complex which gives us the enzyme and the product, and the rate constant is the K2, right, which is the now when there is an inhibitor, the reversible inhibitor, what it will happen, it will bind with the enzyme to form a enzyme inhibitor complex. Just like enzyme substrate complex, we have a enzyme inhibitor complex also, which is noted by EI. Okay. Once the inhibitor occupies the active site of the enzyme, it prevents binding of the substrate to form the enzyme substrate complex, and thus abolishes the formation of the normal metabolic product. The inhibitor binds reversibly the enzyme. Because of that, competition can be decreased simply by adding more substrate. Okay, so how this the reversibility comes, that let's say we have 100 enzyme molecules. Okay, and you are adding up around 50 inhibitor molecules. But if you have more than 50, means if you have, let's say, 200 substrate molecules, what will happen? The rate of binding of the substrate molecule will be greater than the rate of binding of the inhibition molecule. Okay, so this reversibility comes like this. When there is the more substrate can be are added, the competition is decreased, and this reversibly this inhibitor action can be reversed. Okay, so the equation, as I have already mentioned, is this one. So kinetically, the inhibitor I binds with the free enzyme reversibly to form a enzyme inhibitor complex EI, which is catalytically inactive and cannot bind with the substrate. The competitive inhibitor reduces the availability of the free enzyme for the substrate binding. The total enzyme E0 exists in the reaction medium as the free enzyme E, bound enzyme ES, and bound enzyme E0. Okay, so this is a very important. E0 is the total enzyme content. Okay, let us say first we have the total enzyme concentration, which is E0. So this total enzyme concentration is consist of three things. One is the free enzyme, which is not bind with any substrate or inhibitor molecule. Okay, Then we have the ES, which means the substrate bound enzyme molecules. We have EI, means which is the inhibitor bound enzyme molecules. Okay. Excuse me. So in presence of inhibitor, the Michaelis-Menten constant or Km of the normal reaction is increased to new Km. Okay, so this is the new Km, which is 1 plus I concentration of I divided by Ki, which is Ki means the rate constant for the inhibition constant, and then we have the Km where the dissociation constant Ki, Ki is known as a dissociation constant, which is concentration of E multiplied by concentration of I divided by concentration of EI. So the Michaelis-Menten equation for the competitive inhibition, it becomes like this. Okay, we have V0 is equal to Vmax into concentration of S divided by 1 plus the concentration of I divided by Ki into Km plus S. Okay, so this is the in the normal Michaelis-Menten equation is similar to the normal Michaelis-Menten equation where we have V0 equal to Vmax up in the concentration of S divided by Km plus S. But here the Km for the this is the in the presence of the Km, it changes to this form where Km is equal to 1 plus concentration of I divided by Ki into Km, where Ki is the dissociation constant, is which is this one. Okay. Now, consequently, the double reciprocal from or the Lineweaver-Bark equation for is modified as, so if you make the reciprocal of this equation means just you have to make is 1 by V0. So we will have like this 1 plus concentration of I divided by Ki Km divided by Vmax into S plus 1 by Vmax. Okay. So in the presence of the competitive inhibitor, the slope of the double reciprocal plot is increased to 1 plus I by Ki by into Km by Vmax, but the intercept value remains same, which is 1 by Vmax. Okay. So thus the intercept of the x-axis will also differ according to the concentration of competitive inhibitor. So as you can see, this is the Lineweaver-Bark plot for the competitive inhibitor where the y-axis intercept, it is same, which is 1 by Vmax, but the slope changes. In normal enzyme kinetics, normal without any inhibitor, the slope is Km by Vmax, but in presence of inhibitor, the slope is Km into 1 plus I by Ki into by Vmax. Okay. So, however, competitive inhibitors do not affect the turnover number or active site catalysis per unit time or efficiency of the enzyme because of the enzyme is free, it behaves normally. These things are very easy. Actually, it may look like a little difficult, but if you practice when I am uploading the PPT, if you check it directly from the PPT, if you write it few times, you will easily understand and easily remember it. Okay, these things are very easy, but remembering this plot is very important. As I have already mentioned, remembering the plot of different enzyme inhibitions are very important because question can be directly given from the plot also. Okay, and again, mathematical questions are also very important in this type of topics. Then we have the non-competitive inhibition. It is also the reversible inhibition. The non-competitive inhibitor does not have the structural similarity to the substrate, but it binds both with the free enzyme and enzyme substrate complex. So there is a difference. The competitive inhibitor, it was binding only with the free enzyme like this. Okay, this was the enzyme equation. It was binding only with the free enzyme. But the non-competitive inhibitor, it does not have any structural similarity with the substrate. That is why it can bind both with the enzyme itself or with the enzyme substrate complex itself. Okay, so this is the equation, the simplified equation for the steady state kinetics for the non-competitive inhibition. We have the enzyme in presence of substrate, it formed the enzyme substrate complex, and which for dissociated in the into the enzymes and the product. But enzyme can bind with the inhibitor, or should I say inhibitor can bind with the enzyme to form the enzyme inhibitor complex, and this can form enzyme substrate inhibitor complex. This is a new kind of complex which is present in non-competitive inhibition, but it was not present in the competitive inhibition. So this ESI can be formed in two ways. First is the binding of the enzyme with the inhibitor, then binding of the substrate, or second, first binding of the substrate to the enzyme, then binding of the inhibitor. So the presence of the substrate has no influence on the ability of the non-competitive inhibitor to bind an enzyme or vice versa. Its binding alters the conformation of the enzyme and reduces the catalytic activity due to the changes in the nature of the catalytic groups at the active site. EI and ESI complex are non-productive. On increasing the substrate to a saturating concentration does not reverse the inhibition, leading to the unaltered Km but reduced Vmax. Reversal of the inhibition requires a specific special treatment, for example dialysis or pH adjustment, Considering the steady state kinetics of the non-competitive inhibition of the dissociation constant or inhibitor constant Ki of the reaction is this. Follows that the Ki is the concentration of E multiplied by concentration of I divided by concentration of Ei equal to concentration of Es into concentration of I divided by concentration of ESI. Okay, so this is the equation 3. So it is the continuation of the previous equations. So the total enzyme now reaction mixer has a 4 form. So the total enzyme can state as the known the free enzyme, which is the concentration of E. We have the EI, which is enzyme bind with the inhibitor only. We have ES, enzyme bind with the substrate only. We have ESI, where the enzyme bind with the enzyme substrate complex. Substituting equation 1 in the equation 2 is E can expressed as this one. Okay, and then considering the above equation, the Michaelis-Menten equation becomes V0 equal to Vmax divided by 1 plus I by Ki into S by S into plus Km. This is the equation. Says the double reciprocal or Reinweber-Bark plot of the non-competitive inhibitor is given as 1 by V0 equal to Km by Vmax multiplied by 1 plus I by Ki into 1 by S plus 1 by Vmax into 1 plus I by Ki. So here both slope and intercepts are changing. As you can see in the case of normal enzyme inhibitor, normal Michaelis-Menten, the slope is Km by Vmax and the intercept is 1 by Vmax. But here the slope is Km into 1 plus I by Ki by Vmax and the intercept is minus 1 by Vmax into 1 plus 1 by Ki. Okay. So this is the Lineweaver-Bark plot of the uninhibited or non-competitive inhibited enzyme. So these things, these inhibitions and the kinetics, they may seem to be a little difficult at a first glance, but when I am uploading the PPT, you can check it and you have to practice it like equation by equation. Okay, otherwise it will seem difficult. You can practice it today when I will be uploading the PPT, and if you have any problem, let me know by tonight or by tomorrow morning via email. I will repeat it in once more in the next in our last session, which will be on I mean the day after tomorrow, which will be on the Wednesday. Okay, if you have any problem, I will repeat it once more, but first you have to practice it by yourself on a pen and paper when I am sharing the PPT. Okay, these are very easy. I think you can easily you can understand it, because it may seem a little bit difficult, but when you are practicing it by yourself, you will find it is very easy. So this was the non-competitive inhibition. We have the uncompetitive inhibition. This uncompetitive inhibitor has no structural similarity to the substrate. It may bind with the free enzyme or enzyme substrate complex that exposes the inhibitor binding site. It binding, although away from the active site, causes structural distortion of the active site. The allosteric size of the complex enzyme that inactivates the catalysis. This leads to a decrease in both Km and Vmax. This inhibition cannot be reversed by increasing the substrate toward the saturating concentration. Reversal requires a special treatment like dialysis. This type of inhibition is rare and is encountered in multi-substrate enzymes where the inhibitor competes with one substrate S2 to which has some structural similarity and is uncompetitive to other S1. The reaction without the inhibitor will be E plus S1 and ES1 to S2, and EPS with the uncompetitive inhibitor becomes E plus S1, ES1, and then we have this A plus I, ES1I, where prevents S2 binding that produces no product. The steady state kinetics is the uncompetitive inhibitor is as follow: E plus S equal gives rise to ES, which can bind with the I and leads to the ESI, and then considering the steady state kinetics, the uncompetitive inhibition, the dissociation constant Ki is ES concentration into concentration of I by concentration of ESI. Here the total enzyme E0 exists in the reaction mixture 3 form: the free enzyme, bound enzyme like ES and ESI. Kinetically, uncompetitive inhibition and modified Michaelis-Menten equation is this one, where V0 equal to Vmax into concentration of S divided by Ki plus 1 plus I by Ki into S. And then the double reciprocal equation becomes this: 1 by V0 is 1 plus I by Ki by Vmax plus Km by Vmax in 1 by S. Here also the slope is same, Km by Vmax, and the intercept is different, as you can see. So this is the uncompetitive. So remembering the Lineweaver-Bark plots of the three different inhibition, the competitive, uncompetitive, and non-competitive are very important. Okay, if you you do not remember the how it comes, but I mean the final form, try to remember the final form only. Okay, if you do not remember the derivation, I think it is fine, but remembering the final form, the slope, the intercepts are very important, because mathematical question can be given from these topics. Okay. Now, for example, let us see few examples of the inhibitions. So first is the competitive inhibitor. The very important competitive inhibitor examples are the sulfonamides, the simplest form of the sulfa drugs. It is an antibiotic useful in the treatment of some kidney infection. As a structural analog of the para-aminobenzoic acid or PABA. As you can see, sulfonamide is a structural analog for para-aminobenzoic acid. So these are the competitive inhibitors, where the particular where the inhibitor competes for the active site of the our enzyme molecule. Sulfonamide inhibits the bacterial growth. PABA or para-aminobenzoic acid is structural part of the folic acid. Thus, sulfa drug is highly toxic to bacteria that must synthesize their own folic acid. Since human requires folate from dietary source, the sulfonamide is not harmful at a dose that kill bacteria. Methotrexate is another competitive inhibitor for dihydrofolate reductase. The drug closely resembles folic acid and is used as anti-cancer, anti-metabolite chemotherapy, particularly in pediatric leukemia. It hinders the availability of tetrahydrofolate, which is important for anaerobic pathway, particularly synthesis of purine nucleotide for the DNA replication. Examples of non-competitive inhibitors are mostly poisons. Cyanide, azide inhibit the enzyme with iron or copper as a component of the active site or the prosthetic group. For example, cytochrome C oxidase, cyanide binds to the Fe3 plus in the heme of the cytochrome AA3 component of the cytochrome C oxidase and prevents the electron transport to oxygen. Mitochondrial respiration and energy production cease. The cell death rapidly occurs. The central nervous system is primary target for the cyanide toxicity. Acute inhalation of the high concentration of cyanide, for example, smoke inhalation during the fire, automobile exhaust, provokes a brief central nervous system stimulation, rapidly followed by convulsion, coma, and death. Acute exposure to lower amounts can cause lightheadedness, breathlessness, dizziness, numbness, and headache. Cyanide is present in the air as the hydrogen cyanide, in soil and water as the cyanide salt, in NaCN, in food as cyanoglycosides. Okay. Uncompetitive reversible inhibition is rare but may occur in multimeric enzymes. Some example uncompetitive reversible inhibitors are the inhibition of the key regulatory heme synthetic enzyme delta amino levulinate synthetase and dehydratase, and heme synthetase by heavy metal ion. For example, lead, heavy metals like lead from mercaptides such as with thiol in the active site of the enzyme. Okay, now oxidizing agents like ferricyanide also oxidizes thiol into the disulfide linkage. The ability of lithium to exert the profound and selective physicochemical effect has been focus of the considerable research effort. There is increasing evidence that lithium exerts the therapeutic action by interfering with the inositide metabolism in the brain and prevention of the inositol recycling by an uncompetitive inhibition in inositol monophosphate. Few pesticides are uncompetitive inhibitor. The best known example being the herbicide N-phosphomethylglycine, commonly known as the glyphosate, or Roundup, the uncompetitive inhibitor 3-phosphoskipinate or 1-carboxyphenone transferase. Lastly, we have the irreversible inhibition. The irreversible inhibitors have no structural relationship. Okay, let me check if it is fine. Hello. Sorry for the interruption. I think I was disconnected. Is it audible to you? Please let me know if my voice is audible to you. Okay, sorry, there was I think there was a little network issue. So it was fine, right? Up to the example of the different inhibitors, it was fine. Just let me know when it got disconnected. So up to the example of different inhibitors, was it fine? Just let me know where I just let's do it. I will share the PPT. So up to which slide it was fine? So up to this slide it was fine, right? Please let me know if it was fine up to this slide, or should I repeat this slide? Okay, I think up to that slide it was fine. Then it got disconnected. So I can start with the irreversible inhibition. Okay. So the irreversible inhibition have no structural relationship to the substrate, and it can bind covalently with the active site of the enzyme and destroy the essential functional group of the active site. So irreversible inhibitors are used to identify functional group of the enzyme active site at the location they are bound. Subsets of the irreversible inhibitor called the suicide irreversible inhibitor are namely inactive compounds. They get activated upon binding with the active site of the specific enzyme. However, it does not release any product because it irreversibly binding with the enzyme active site. The enzyme, in fact, commits suicide by activating the inhibitor. Due to this very nature, suicide irreversible inhibitors are also called the mechanism-based activator or transition state analog inhibitors. The transition state analogs are extremely potent and the specific inhibitors because they have higher affinity and stronger binding to the active site of the target enzyme than the natural substrate of the product. However, exact design of the drugs that precisely mimic the transition state is a challenge because unstable structure of the transition state enzyme catalyzed reaction. Kinetically, the irreversible inhibitors decrease the concentration of active enzyme and in turn decrease the maximal possible concentration of enzyme substrate complex. It reduces the initial concentration of the enzyme E0 until all the enzyme are consumed to E0 and I0 level. Once the process between the enzyme inhibitor gets completed, no free enzyme is present in the mixture. Further addition of the substrate follows the Michaelis-Menten equation. In absence of the inhibitor, the rate of the formation of product from the excess substrate Vmax equal to K cat is 0. While in presence of inhibitor, the rate of the excess substrate formation it's the formation of the product from the excess substrate is Vi by max in kcat into E0 minus I0, where kcat is a turnover number which is defined as the maximum number of molecules substrate enzyme can convert in per catalytic site per unit time. Combining the above two equation gives us this, which turn reduced to this. This Vmax reduced to Vi max in presence of irreversible inhibition. That is similar to the reversible non-competitive inhibition, but in irreversible inhibition, relationship between Vmax and Ivmax does not involve the Ki. Some clinical example of suicide inhibitor is NN-dimethyl diethyl propylamine. Okay, the common compound inhibit the enzyme monoamine oxidase or myo. Myo is responsible for breaking down the neurotransmitters such as dopamine, serotonin, thus decreasing their concentration. Concentration in the brain. Diseases such as Parkinson disease, depression occur because decreased level of the dopamine and serotonin. Then I have mentioned that remember you can read about all the purification techniques because short notes can be given on the different purification technique. For example, gel filtration chromatography. So chromatography encompasses a variety of the techniques that involve a mobile phase flowing through a bed of the stationary phase. A mixture of solute that is resolved and separated as a result of such differences in affinity of the solutions for the mobile phase relative to the stationary phase. A solute that has more affinity to the mobile phase move forward with the mobile phase, whereas the solute with more affinity for the stationary phase is retarded. The solutes are separated into bands and the distribution between the phases. Molecules of biological interest is separated or resolved by the variety of chromatography technique, including the adsorption chromatography, ion exchange chromatography, partition chromatography, or paper or thin layer chromatography, gas liquid chromatography, molecular exclusion chromatography, and molecular C1 gel filtration. Chromatography involves a mobile phase, liquid or gas flowing over a stationary phase, solid or liquid. This is the example of column chromatography where we have a stationary phase. This is the round shapes and M is the mobile phase. Okay, then a mixture of solute is separated based on their solid components' degree of affinity on the mobile and stationary phase. For example, if component 1 has a stronger affinity of the stationary phase than component 2, then 2 will migrate through the column more rapidly than 1. We have the size exclusion chromatography where the stationary phase have the different size of pores, and this chromatography or the separation happens when the larger molecules are smaller molecules. So if the molecules are smaller, it can enter the pores of the stationary phase and bind more strongly with the stationary phase, whereas the larger molecules are unable to bind with the fit into the pores, and they will separated at first. Okay, so this is the size exclusion chromatography. Then we have the purification of proteins by the ion exchange chromatography. So if the column is negatively charged, for example carboxymethyl, then the protein with the Pi less than pH buffer will be negatively charged and not bind to the column and elute. Proteins with the Pi greater than the pH of the buffer will be positively charged and it will be bind to the column. This is the simple, I think I have discussed it earlier in one class that when the proteins have Pi less than pH of the buffer, it will be negatively charged, and when the Pi's are greater than pH of buffer, it will be positively charged. So now this the technique is similar and simple. Now if the column is negatively charged, for example like this, and if you have the protein which are positive or negative charge separately, so the protein which has the positive charge, it will bind easily, and okay, and then if it has the sample loading like this, positively charged protein will immobilized on the anion surface and eluted with the salt gradient of NaCl, and with the low salt gradient like NaCl, but when the negative charge, they will be eluted at the high salt gradient. Okay, we have the 2D gel electrophoresis. Okay, so in the 2D gel electrophoresis, proteins are separated as per their isoelectric point and protein mass. So it is known as 2D because there are two different separation. First, based on the decreasing Pi, and the second is decreasing the molecular weight. Separation of the proteins by isoelectric point is called the isoelectric focusing. When the gradient of pH is applied to a gel and the electric potential is applied across the gel, making one or more positive than other, at all the pH values other than their isoelectric point, proteins will be charged. If they are positively charged, now they will be pulled toward the negative end of the gel, and if they are negatively charged, they will be pulled to the positive end of the gel. The proteins applied to the first dimension will move along the gel and will accumulate at the isoelectric point, that is the point which is overall charge as the zero. And separating proteins by mass is the gel treated with the SDS along with the other reagent like pH. So, and denatured proteins, that is unfolded into a long straight molecule, binds the number of SDS roughly proportional to the protein's length. Because the protein's length when unfolded is roughly proportional to its mass, since SDS molecules are negatively charged, result of the gel proteins will have approximately same mass to charge ratio as the other. Okay, so this is the 2D gel electrophoresis. Now the last topic which I would like to focus on a little bit is the cholinergic system. So somebody has asked to repeat this thing because I have not discussed it in detail. Only I have given a question in a particular in a specific week. I don't remember the exact week. So let us discuss a little bit about the cholinergic system. So the cholinergic signaling system. So first, now let us look at what happens in this figure. Okay, this now what happens at the synapses involving acetylcholine as the neurotransmitter. So this inverted triangle is the acetic acid. Then this rectangle is a choline. This shape is the acetylcholine, and E1 is the acetylcholine acyl transferase. Okay, and this is the post synaptic nerve, and this is the presynaptic nerve. So what happens? it shows that the synapse between the two neurons and the events involved when a message is transmitted from one neuron to other. The same general process takes place when a message is passed from neuron to muscle cell. The first stage involves the biosynthesis of acetylcholine, which is this one. So how acetylcholine is biosynthesized? We have this choline, and then this is the structure of choline, and we have the acetyl-CoA. Okay, so acetylcholine is synthesized from the choline and acetyl-CoA at the end of the presynaptic neuron. So here, the first is the acetic choline and acetyl-CoA. Now, okay, both forms this acetylcholine, and which is in which stays and in a vesicle. So acetylcholine is incorporated into the membrane-bound vesicles by the means of the specific transportation protein. Then the arrival of the nerve signal leads to an opening of the calcium ion channel. This is the opening of the calcium ion channel. Okay, now this opening calcium channel and an increase of the intracellular calcium concentration. This induces the vesicle to fuse with the cell membrane and release the transmitter into the synaptic gap. This is the synaptic gap. The acetylcholine crosses the synaptic gap and binds with the cholinergic receptor. So here in the postsynaptic cleft we have the receptors which is known as the cholinergic receptors, and acetylcholine when after crossing this gap will bind to this cholinergic receptor. Okay, and resulting the stimulation of the second neuron. The acetylcholine moves through an enzyme known as the acetylcholine esterase, which is situated in the postsynaptic neuron. Okay, in the postsynaptic neuron there is an enzyme known as the acetylcholine esterase, which catalyzes the hydrolysis of acetylcholine to produce the choline and acetic acid. Choline is taken up by the presynaptic neuron by the transport protein to continue the cycle. So this is the overall cycle. Okay, so here this particular acetylcholine is taken up by the cholinergic receptor, which is present in postsynaptic cleft. After that, it cleaves. There is a cholinergic acetylcholine esterase is present, which will cleave this acetylcholine esterase to choline and acetic acid. Choline is recycled back to the post presynaptic nerve, where it will again used to form the acetylcholine. So key point, the cholinergic nervous system involve the nerves which use the neurotransmitter acetylcholine as a chemical messenger. These include the motor nerves which innervate the skeletal muscle and nerves which synapse with other nerves of the peripheral nervous system or PNS, and the parasympathetic nerves innervating cardiac and smooth muscle. There are two types of cholinergic receptor, muscarinic receptors which are present in the smooth and cardiac muscle, and nicotinic receptors are present in the skeletal muscle and synapses between the neurons. Acetylcholine is hydrolyzed by the enzyme acetylcholine esterase when departs from the cholinergic receptor. The hydrolytic product of choline is taken up by the presynaptic neurons and related back to the acetylcholine. The cholinergic receptor of the enzyme acetylcholine esterase is useful drug target. Acetylcholine cannot be used as a drug because rapidly hydrolyzed by acid and enzyme. It shows no selectivity for different types and subtypes of cholinergic receptors. And these are the different type of cholinergic receptors. So cholinergic receptors can be two type. One can be ligand gated ion channel, and then we have the G protein coupled receptors. Ligand gated ion channels is nicotinic acetylcholine receptors or NASCHRS, and G protein coupled receptors can be like muscarinic acetylcholine receptors, which are 5 type M1, M2, M3, M4, M5. Nicotinic receptors are mainly of two types. One is the central nervous system plus autonomic ganglia in peripheral nervous system, which is the neuronal NSHRS, and we have skeletal muscle in neuromuscular junction, which is the muscle NRS, which can be 5 types: alpha 1, beta 1, gamma, delta, and epsilon. The neuronal ones can be two type: homopentameric and heteropentameric. Homopentameric are alpha 7 and alpha 9 are two type, and heteropentameric combination of alpha beta subunit and different alpha subunits. For example, this one. And neuronal subunit can be of alpha and beta subunits are different. So alpha subunit is like alpha 1 to alpha 10. Alpha 1, 3, alpha 2, 3, 4, 5, 6, 7, 9, 10, and beta 2, beta 3, and beta 4. Okay. So these are the different type of the cholinergic receptors. So this was the last slide of today's session. So today what we discussed, we revised the topics which we covered till our last 12 weeks, and also we discussed about the kinetics of different enzyme inhibition, and followed by cholinergic receptors, separation, purification techniques, and amino acids a little bit. So this is my email ID which I will share now. If you have any doubt, any problem, or you want me to want me to cover up any topic in our last class, which will be on 16th October, let me know by tomorrow morning. Okay, I will try to include the slides. Otherwise, I will just make the revise. So in the last session, it will be 1 hour summary session. I will not give any question answer in that day. I will only discuss the topics which are very important in this course of organic chemistry and biological and drug development. Mainly the topics will cover the different agents like anti-cancer, antibacterial, antiviral, and so on. Okay. So thank you for joining. I will leave the session today. So we will meet on our last live discussion session on 16th October from 7 to 8 p.m. Okay. Thank you all for joining. I will close the session for today.